Good evening. This is Dr. Dan Guerra from VerEvMed, and once again we are talking about aspects of biomedicine, and this is a lecture that I have promised we were going to do today. This is the day after Thanksgiving 2017. So let's get right into it. Okay, so remember this is VerEvMed, and I'm the Chief Scientific Officer of this company. And what we do is bring the scientific literature out in the open, and we discuss it, we vet it, and we compare it to the other literature and try to come to some conclusions as to how this new information that comes out every day can be associated with all the other material that we understand in a given topic. So recently I've been talking about apoptosis, that is programmed cell death, autophagy, or that is the specific utilization of intracellular organelles and polymeric structures in order to maintain solvency in a cell so it doesn't have to take terminal cell fate, and senescence, that is the aging of cells, ultimately leading to a lack of reproduction, that is division, and then ultimately to death. So we've been embedding that general cellular phenomenon with the discussion of cancer. So we're going to continue along that arc. And today I'm going to talk to you about anti-angiogenesis. Angiogenesis, of course, most of you know, simply just means the synthesis of new blood vessels. So let's get started. Again, this is my photograph. This was taken very recently out in the Clearwater National Forest, St. Joe National Forest. This is a load of wood I picked up, and if you look at this wood, you can tell this is actually fir, white fir. And this is section 13, where I do a lot of wood cutting in that region. I know that matters to a lot of people. So again, the title of the talk today is State of Anti-Angiogenesis in Cancer Pharmacotherapy. A subtitle is Blocking Blood Vessel Development May Not Be a Uniform Mechanism to Halt Tumorigenesis. Okay, so let's get into this whole topic. First of all, let's take a look at a recently published paper about a year ago in a journal called Glioblastoma. Let's go through what it says. It's the summary or synopsis of what this paper said, and I thought it was a good introduction. So they tell you that in something we know, a major mechanism for anti-angiogenesis therapy targets a specific protein called VEGF and its receptor. VEGF is vascular endothelial growth factor. The therapeutic strategy serves to minimize tumor-associated blood vessel production. So that's what it means to be anti-angiogenic. The result, of course, is considered to limit nutrition to the developing tumor and coincidentally to help control targeted drug delivery. That's by controlling basically the vascular beds. Furthermore, a normalization of angiogenesis, which is what we mean when we target angiogenesis in proliferating tumors, we're basically normalizing angiogenesis. Anyways, that could limit intratumoral hypoxia by instantiating short-term perfusion. Now, hypoxia is what happens when you have a decrease in blood flow, right? You get also a decrease in molecular oxygen, and that's what hypoxia is. Now, when a cell is delivered that fate, it turns on a whole repertoire of genes generated by the hypoxia induction factor, HIF, and HIF then induces the cell to reprogram itself and survive that low oxygen tension. And that's part of actually the pathology that promotes tumorigenesis, okay, sort of paradoxically. Okay, so let's go beyond this now. The main paper I'm going to be delivering from is Chronic Disease Translational Medicine, published March of this year. So let's take a look at what this paper wants to tell us in the introductory remark. Again, anti-angiogenesis therapy is designed to inhibit the formation of new blood vessels in tumors. It's a standard of care therapy, actually, for many cancers. For those of you that are on any kind of pharmacotherapy, you are aware of this. And of course, the physicians in the audience are as well. Basically, it's a mechanism, as we've said, to starve the tumor and lead the cell mass into some terminal fate. So senescence, so that is the aged cell, no longer can divide. Obviously, that's a good phenotype for a cancer cell. But it can also lead to apoptosis, programmed cell death. And as we'll see, it can also lead to autophagy. Now, those three different cell fates all have a different result when you are doing them in tumor cells. So what if autophagy is the result? Okay, so let's recap what we mean by that. Sorry for all the writing, but I think it's necessary for us to get through this because I think many people do not have a full grasp on what autophagy is. So I want to put this on my slides so you can read them even when I'm not reading them to you. So what is autophagy? It involves the partitioning and reorganization of the cell. It leads to a selective turnover and reorientation of non-nuclear organelles like the mitochondria, the endoplasmic reticulum, Golgi apparatus, and the peroxisome. It also causes a turnover of protein aggregates and filaments, that's part of the cytoskeleton, and nucleoprotein complexes. The substrates for autophagy include the entire organelles, as I said, also massive amounts of cytoplasmic inclusion bodies, such as endosomes, cytoskeletal glycoproteins, polymeric protein complexes, and soluble signaling proteins and even metabolic enzymes. And what you put all of that into is a newly formed structure called an autophagolysosome, which we'll see a cartoon of in a moment. Ultimately, sections of the autophagous subcellular structure can be partitioned by specialized phagophore membranes. So membranes are laid down within the cytoplasm. And what happens when you do that is you digest the material inside that, limited by that phagophore membrane. And you yield sometimes needed respiratory intermediates directly for bioenergetics. You can also break down to precursor compounds for biosynthetic metabolic needs, such as amino acids. So autophagy can be induced by preventing import of nutrients, so starving, or blocking secretion of waste products or buildup of toxic compounds from subcellular compartments, whole tissues, and isolated cells. The process is also linked to differentiation. It's not just when a stress phenomenon occurs, and normal development, as you might guess. And it's believed to be a programmed response to achieve homeostasis of individual cells within a tissue phenotype. Here's the cartoon I promised you. So let's take a look first at when we get actual autophagy. So nutrient depletion and stress can do this. And also, as we will see, anti-VEGF pharmacotherapy, which is going to be the main topic here today. That's going to turn on this enzyme, AMP kinase, which I've talked about several times in various other VerEvMed lectures. For right now, just so you understand, this is induced, AMP kinase is induced by adenylyl monophosphate, and it's a phosphorylating enzyme. So it binds AMP, turns on this kinase, and first of all, for purposes of this discussion right here, it blocks the mTORC pathway. That's targeted rapamycin. Now, the mTORC pathway normally would be involved in biosynthesis of proteins, and basically when the cell is flush, mTOR is turned on. So AMP kinase is going to shut that down. When you shut down mTORC, you shut down this. mTORC normally shuts this down. When mTORC is shut down, this is allowed to proceed. All of these proteins are allowed to aggregate, pick up this ATG9L, form this complex here known as the phosphorylanositol 3 kinase 3 complex. That then generates, okay, because it's a phospholipid, so it's going to start causing an aggregation of phospholipids into what's called a phagophore. Phagos means, of course, to eat, right? Phagos. It's going to pick up all of these proteins here, ATG proteins. That's then hierarchically going to allow the further accumulation of the LC3 proteins with more ATG proteins now assembling into an isolation membrane with cargo. You see what the cargo is. Those are the organelles in the cell, and again, these polymeric protein structures. One more protein then is associated. It's the same one that aggregated here, LC32, and now you close that, you limit that structure, and you make an autophagosome. That now is going to deliver to the lysosome, and you're going to make an autophagolysosomic structure that's going to allow the complete, because the lysosome is going to have all the enzymes that are going to allow you to degrade, so phospholipases, proteases, nucleases, enzymes like that, glycosidases, and you're going to break down all of the contents that's inside that autophagolysosome, going to degrade it down. So that's what we mean by autophagy. Now, when you have growth factor stimulation or insulin stimulation, normally you turn on another phosphatidylanosyl cascade system, turns on another a protein kinase, PDK1, which turns on AKT, which turns on mTORC. And what mTORC does is block this whole thing. So when you have a well-fed state, such as high circulating insulin or growth factors like VEGF, what it does is it blocks all of this system. Okay, so you do not get the autophagy turned on. So that's what. So when you use anti-VEGF pharmacotherapy, basically this is the target for that particular drug. It's going to be targeting either VEGF binding to its receptor or the receptor itself. So right away, I want to explain, because this is what I've been doing, is that in all of these pharmacotherapies for cancer, the hallmark is that we generate resistance. It doesn't mean that the drug no longer has any opportunity to be bioactive. It simply means that when you constantly select for cells by using a very potent drug, like an anti-VEGF drug, you're going to then isolate some of those cells that become resistant. So when you get resistance to this, what looks like a very potentially very good anti-tumor drug like VEGF, because, again, we're fighting tumors here, because you're blocking angiogenesis by blocking VEGF. And what that does then, as you know, it depletes nutrition to those tumors. How does the resistance happen, right? So I'm going to show you some of the mechanisms. So, okay, start off here. You've got a vascularized tumor, means you've got a lot of good blood vessels that are feeding that tumor mass, okay? That's the hallmark of what a tumor looks like, right? You add the anti-angiogenic therapy like anti-VEGF. And what happens is you get a reduction and normalization of the blood vessels, okay? So you don't get any new blood vessel development. Now, that is what you want, right? Because the less nutrition, that means that these cells, these tumor cells can't divide. However, where are the resistance mechanisms and how do they occur? First of all, you can have alternative pro-angiogenic pathways. So besides the one you targeted, like VEGF, there are several other ones, including FGF, PDGF, EGF, HGF, Notch. So there's a series of other possible angiogenic pathways, not just VEGF, shown down here, of course. That's one method, okay? So there are backup supply to be able to maintain this vascular bed genesis. Second way is neoangiogenesis, independent mechanisms of blood vessel formation. Here, the endothelial cells start turning on expression of genes so that they start acting like tumor cells. And when that happens, the endothelial cell no longer is controlled by the anti-angiogenesis factors. Okay, remember the endothelial cells, what you're going to be is going to be controlling where that vascular bed is laid down. That's the second mechanism. Third mechanism, basically just an escape from hypoxia. So you get vascular co-option, invasion of healthy tissue. So you just avoid, so the tumor just avoids that portion of the tumor, maybe the center of the tumor, where the new vascular beds were laid down. And just wherever that does not happen, new vascular beds are going to be laid down proximal to wherever that region was that was hypoxic. Fourth possibility is involvement of tumor-associated stromal cells. So these are also known as tumor stem cells. So you get a recruitment of CD11B+, GR1+. These are proteins that phenotype turns up in the myeloid lineage, okay, directly from the bone marrow. and that promotes angiogenesis directly. So what you get then is basically entirely new cell structures coming to the scene, feeding into that tumor mass. Finally, you get tumor cell adaptation, and one of the adaptations that actually allows for resistance is autophagy itself. Okay, that's a way of dealing with it, because remember, autophagy doesn't allow the cell to take on a terminal fate. Autophagy is like regrouping the cell and allowing it to deal with this selection pressure, this anti-angiogenesis. So all of these possible resistance mechanisms, or any one of them, can make the tumor itself, the tumor mass, resistant to your standard anti-angiogenic therapy like anti-VEGF. So when VEGF is working, it requires both the VEGF protein and the receptor. So not only do we have immunotherapy that blocks the VEGF protein from binding to its receptor, we also have inhibitors of the receptor itself, okay? So the growth factor receptor inhibition is what we're talking about here. And these are going to be receptor tyrosine kinases. That's the kind of receptors you have. So there's a compound called sunitinib. Now, what sunitinib does is block the phosphorylation of these tyrosine kinase receptors, which VEGF is one type of receptor that's like that. So if it blocks that, you're going to decrease AKT and ERK signaling, okay, which normally what VEGF would be doing. And when you inhibit that, you inhibit cell proliferation. So this is going to be a cell that's now sensitive to, here's another drug, it's an inhibitor drug. You know it's an inhibitor because it has the IB ending. When you use an immunotherapeutic, it's going to be an AB ending for antibody. Okay, IB is inhibitor. Now, what happens is that in some cells, because you're pushing the selection pressure on the cell mass, this tumor cell mass, this sunitinib actually can pharmacokinetically be taken out of its normal inhibitory state and sequestered in the cell. So if it's sequestered in the cell, for example, in an endosome, it no longer can function, the drug can't function, or maybe it's sent out of the cell via an ABC type of translocator, or maybe the compound actually, the drug can actually be broken down in the cell. At any rate, what happens is you do not block the phosphorylation. You get complete phosphorylation of the receptor tyrosine kinase. You get an uptick in signaling, and you get an uptick in cell proliferation to compensate for that initial phosphorylation inhibition. So now you got a resistant tumor cell to this compound that was supposed to be blocking the receptor tyrosine kinase. So it's another whole pathway, not just the VEGF protein itself, but the VEGF receptor. So let's take a look at this and put this together. So this bevacizumab is a monoclonal antibody to the AB that normally blocks not the receptor, but the VEGF protein itself, just showing you blocking your VEGF isoform A. Now, one thing to remember about growth factors is that they act in an autocrine way. That is, they're synthesized in the cell that they are now going to bind back on a receptor in that same cell. That's the autocrine activity, okay? Autocrine hormones are hormones that are synthesized within the cell that they're utilized in. So you got the receptor in the same cell where VEGF is now secreted, comes back in. Now, because it's an autocrine regulation, that means that the expression and the secretion of the VEGF protein, and then the binding as ligand to its receptor, is going to be associated with the VEGF receptor dynamics. That is the expression of the receptor as well, okay? Now, when you block that with the monoclonal antibody, you block the autocrine mechanism. So what happens is the VEGF receptor now becomes starved to the ligand. So one thing the tumor can do is increase the expression of the VEGF receptor, okay? So this seems paradoxical, but it's not really, because this is how receptor-mediated endocytosis works. When you have a ligand binding to a receptor, it doesn't matter what it happens to be, when you inhibit the ligand concentration by blocking it with a monoclonal antibody, the cell responds because remember, it's trying to synthesize this. It's synthesizing a lot of VEGF. It's a tumor cell, so it's trying to lay down new vascular beds. All it's going to do now in response is make more of the receptor. So whatever VEGF is there will be scavenged because you're going to have more receptors, and now you're still going to get a response of angiogenesis, right? So VEGF receptor expression gets upticked. Now, what else happens is a protein called PIGF expression also gets turned on. Now, what's PIGF? Thank you for asking that question. PIGF is phosphatidylinositol glycan biosynthesis class F protein. Now, phosphatidylinositol is a phospholipid that is used to anchor proteins to the plasma membrane. Okay, what kind of proteins? Proteins like receptor tyrosine kinases, specifically the receptor tyrosine kinase VEGFR. So when you make more of this enzyme, you're going to make more of this GPI, this glycosylphosphatidylinositol anchor. That anchor is going to be facilitating whatever VEGF receptor is now being uptick synthesized, that is transcribed, translated in this tumor cell, so that now you're also going to enhance the availability of the receptor on the plasma membrane surface because you've made the GPI anchor. So this protein now is going to be out there binding whatever VEGF-A is available that's going to overcome the monoclonal antibody, suppression of its activity. So the cell's very sneaky. The cell's going to enhance the expression of the receptor. It's also going to enhance the expression of enzymes that maintain the receptor's viability on the plasma membrane itself, right? By adding this GPI anchor. So reinforcing its anchoring on the membrane makes it more available, more transparent to whatever VEGF-A is now available. So you're going to, again, paradoxically, you're going to get a pro-tumoral effect, right? You're going to get more angiogenesis, right? Likewise, the cell is also going to start turning on the downstream proteins that normally will be stimulated by VEGF. And so the tumor cell is going to make more AKT and ERK, and that means you're going to move more towards the mTOR pathway, more towards cell division, okay? So the GPI anchor, a glycolipid containing 3 mannose molecules, that's giving you some detail about what it is. The encoded protein, another GPI synthesis protein called PIGO, function in the transfer of ethanolamine phosphate to the third mannose of GPI. So all I'm giving you here is the detail of when you uptick PIGF, you've got this whole biosynthetic machinery is now enhanced, right? And because it's enhanced and enabled, all it's doing is building up this VEGFR reception, so that again, you get sort of a paradoxical effect. And this is shown in glioblastoma. And I got this whole diagram from a paper in Transit Molecular Medicine. Okay, so let's now do a history of why we even do anti-angiogenesis. We've seen now how resistance can occur. Let's talk about why this was afforded in the first place as an idea to block tumorigenesis. Again, a lot of writing. I want to put this out there so you can go back and review these slides. So solid tumor progression requires the genesis of new vascular beds, as we've said. Okay, it gives you oxygen, it gives you combustible carbon, right? So bioenergetics, it also gets rid of the waste of the cell. The central target for pharmacological treatment of tumors is the molecular blockade of VEGF, because VEGF is a growth factor. What's it a growth factor for? Angiogenesis, okay? So anti-VEGF drugs are significant in armamentarium for anti-angiogenesis oncologic pharmacotherapy. Okay, that's chapter and verse. Now, where does this come from? Wasn't that long ago, in 1971, Volkmann and colleagues observed that tumors were restricted to very small size until neovascularization was introduced. So this began the anti-angiogenesis anti-tumor pharmacological campaign. So bevacizumab targeting VEGF proved very useful in metastatic colon cancer. All this was all good 14, 15 years ago. Then it was a few years later, non-small cell lung carcinomas in 2006, and advanced cervical cancer in 2014 were also benefited by the monoclonal antibody that blocks VEGF. Okay, so that's what this compound is here, right? This bevacizumab is a monoclonal antibody that's blocking the VEGF protein, right? Binding to it. Simultaneously, these inhibitors came on the scene way back in 2005. Sorafenib came on. Again, it's a multi-tyrosine kinase inhibitor. We call these RTK small i's. Those antagonize the VEGF receptors, okay? And so progression-free disease beat the placebo. Okay, so what this sorafenib showed is that you got a decrease in progression of the disease, okay, against a placebo, which did not have any drug at all, in advanced renal carcinoma. So that was back in 2005. So that was a very good data, okay? So what that data said was, yeah, we did not progress with this particular really nasty advanced renal carcinoma, kidney disease, kidney cancer. And so a whole family of other RTKIs were synthesized and tried out. However, the monoclonal antibodies, like the one that blocks the VEGF, okay, and all the RTKIs that are blocking now the reception of these growth factors, only seem to benefit when combined with conventional chemotherapy, okay, the normal drugs that are thrown at tumors. And even this was met with inconsistencies and resistance in randomized controlled trial settings. And overall, okay, in the meta-analysis of the data now in 2017, clinical evaluation was not overly optimistic. So not only did the monoclonal antibodies not work as a monotherapy, and the receptor tyrosine kinase does not work well as a monotherapy, even when they're combined and even with conventional chemotherapy, when you look at all the trials have been where these have been set up and used, we just don't have a really good, solid decrease in these cancers. So what that means is you're building up resistance, right? Okay. So we might ask a little bit more detail of what's going on inside the tumor. Why doesn't the anti-angiogenesis strategy work all that well? Okay, so it's like pulling back out, just asking a general question, again, using categorical logic to answer these questions, and then going back into the literature and examining how this was answered. First of all, anti-angiogenesis strategy only blocks new blood vessel formation. The older mature blood vessels are fine. We're not damaging those, and those can actually keep a tumor bathed in circulation. That's one thing, okay? Second, when vasculature is restricted, such as using these anti-angiogenesis compounds or strategies, the tumor just switches metabolism to the anaerobic mode. It's again now going to express the hypoxia-inducing factor, HIF, and that is going to be a transcription factor, which is going to turn on a series of genes in the tumor that's going to allow it to survive this period of hypoxia, okay? It also allows it to, because of the proteins that are going to be synthesized by the HIF acting as a transcription factor in the nucleus, are going to actually allow the tumor now to evade the immune system. So what this all can do when HIF is turned on, because you're blocking this de novo angiogenesis, right? It selects for tumor stem cell formation. So now you're going to start making new tumor cell masses, because you're going to be converting the normal cells now to tumor stem cells so they can take any fate they want. It also induces the HIF system, the programmed death ligand expression from the tumor. So when that ligand binds to the programmed death receptor on cytotoxic T cells, it causes the T cells to kill themselves. Okay, so this is a way of not only evading the immune system, but eliminating the immune system. Because remember, the cytotoxic T cells are going to be mobilized to that tumor. And here we go. The tumor is going to make this ligand. The ligand is going to bind to receptors on the T cells. And it's going to kill the T cells. So it's not a good thing. So inducing HIF can cause this particular response. So ultimately, a pharmacological pseudo-paradox is obtained. Pro-VEGF, pro-VEGF actually switches back to normal angiogenesis that shuts down the HIF system, reestablishes conventional drug delivery to the tumor. So interestingly enough, when a person's on an anti-angiogenesis drug for a while, they may be weaned off of that drug, okay? They may be completely tapered off that drug once the HIF factor starts to generate a tumor mass that's now evading the immune system, and also worse than that, it's killing immune cells. So get rid of the anti-angiogenesis compound, the drug, and go back to allowing VEGF to be expressed. So now you can hit this tumor with the old chemotherapeutics, or maybe go after it with radiotherapy or surgery, of course, too, which is, of course, always a possibility, always a therapy. So what this tells us is that in these tumors, it's a very complicated system. There's a tissue-specific timing of VEGF expression, and it has to be an important consideration when using anti-angiogenesis pharmacotherapy, such as the anti-VEGF. So VEGF can promote senescent cell transitions, thus leading to tumorigenesis later in life. So we have to be careful about when we go after targeting VEGF, okay? Because when you target at the wrong time, earlier in the stages, maybe even of the age of the patient, it's going to have a different effect than when that patient's older, okay? And we're going to see what I mean by that in a moment. And in particular, epithelial cells can transition to mesenchymal cells. That's called epithelial mesenchymal transition upon VEGF signaling. So this is a very bad thing. Mesenchymal cells are like stem cells, okay? It's putting them back into a more primordial, more juvenile cell state. So now they can go and they can turn into tumor cells, right? Once these epithelial cells have made this transition. And VEGF will do that, okay? So again, using anti-VEGF and then getting a boost of VEGF expression because of the anti-angiogenesis drugs early on in a pharmacotherapy of tumors can actually result in another entire system, not in the tumor cell mass, but just in the epithelial cells associated with it. And you get this EMT effect transitioning to mesenchymal cells. So let's take a look at this. Now I'm going to explain to you what I mean by that. The epithelial mesenchymal transition, EMT, is a biological process that allows a polarized epithelial cell. These are normally polarized cells. They normally interact with basement membrane, so they're polarized to that via its basal surface, so that they undergo multiple biochemical changes that enable it to assume a mesenchymal cell phenotype. When this happens, it includes enhanced migratory capacity, invasiveness, elevated resistance to apoptosis, and greatly increases the production of extracellular matrix components, okay? And also it allows for extracellular matrix melting so that now that invasive process can proceed, okay? So you're making extracellular matrix degrading enzymes, okay? So this is all bad, right? This is all a terrible thing to happen when you make this transition. And I'm telling you that VEGF can turn this on. And in fact, look at this phenotype. Enhanced migratory capacity of what were once epithelial cells, okay, detaching. They can invade, and they're also resistant, okay, to apoptosis. What does that look like? That's a tumor cell, right? Yeah. And likewise, breaking down extracellular matrix. So the completion of an EMT is signaled by the degradation of underlying basement membrane, so they detach. Formation of the mesenchymal cell now lineage can migrate away from the epithelial layer in which it originated. This is all very bad news, okay? So these are distinct molecular processes, and they're engaged in order to initiate an EMT, okay, a mesenchymal transition, including the activation inside those cells of transcription factors, expression of specific cell surface proteins, reorganization, expression of cytoskeletal proteins, production of extracellular matrix degrading enzymes, and changes even in the expression of so-called microRNAs, which actually serve normally to prevent this from happening, okay? So when you get expression of a specific set of microRNAs, they're going to now, they can be exocytosed, and they can further control the transition in other cells associated with the ones that have already made the transition. So very bad news, right? So here are the observations. Cancers of epithelial origin, which are carcinomas, are the most frequent type of malignancy in humans, okay? They have high incidence and they're aggressive, and all of that increases with age. The observation raises the question as to what is aging doing that contributes to this? Well, there are a couple of things to consider. And we talked about this in previous VerEvMed lectures, and I'm just reiterating it now in case you haven't watched those lectures. First of all, telomere biology seems to play a pivotal role since shortening of those telomeres has been associated with cellular senescence and organismal aging, as well as with cancer incidence and mortality. Now, how does that make sense, okay? Well, in the multi-step carcinogenesis model, telomere shortening has been observed together with something called chromosome instability overall. And chromosome instability, or CIN, and when you add telomerase, it's TCIN, that's the acronym. That's a precancerous condition. Okay, so I'll explain to that in a minute how that can happen. Because remember, telomere shortening basically gives you a shelf life of every cell. Telomere is that extension of the chromatin. It keeps on getting shorter and shorter after every cell division. Eventually, telomerase, which adds this telomere, this piece of DNA to the ends of chromosomes, the telomerase enzyme starts to decrease in expression. You make less and less of the telomere, less and less of its made. Eventually, the cell becomes senescent. That's supposed to happen, okay? But the telomere itself, its instability, can be followed by a telomerase reactivation, okay? And if that telomerase is reactivated in otherwise quiescent senescent cells, you can get that metastatic phenotype. This is happening regardless of any tumor growth, regardless of any drugs that could be attacking to. This is what happens when you age. This is why aging is associated with carcinogenesis, okay? Because this telomerase decrease in expression, telomeres are getting shorter. What can happen is a superinduction of telomerase, and then that cell lineage, because it now already has picked up these mutations, can then result in a metastatic phenotype. So that allows me to now talk again about something I've discussed in previous VerEvMed lectures. These are called SASPs, okay? Now, SASP is the senescence-associated secretory phenotype. So when you have a senescent cell, sometimes senescent cells can become SASP phenotype. And when that happens, it's a very bad thing. Let me explain why. So telomere-driven chromosome instability, or TSIN, as I told you we were going to call it, in human epithelial kidney cells induces a global microRNA deregulation and the acquisition of phenotypes, which then cause a metastatic capacity or capability. While that's going on, meanwhile, the senescent cells display the so-called SASP. So you got chromosome instability because of telomere shortening that induces a microRNA deregulation. And when that happens, you get an alteration in gene expression, because that's what microRNAs do, okay? Because remember that microRNA binds to a target RNA and pulls that target, if it's a messenger RNA of a particular gene is being expressed, it pulls that expression out of the mainframe of what the cell's fate's going to be. So if you make an alteration in the microRNAs, you're going to alter cell fate. Okay, and that's what telomere shortening does, and that's why you get chromosome instability. Okay, so protein going back and feeding into chromosome instability. So senescent cells, when they turn on the SASP phenotype, what that phenotype is secretion of a large variety of cytokines, okay, which can be pro-inflammatory, and also, what's this? Growth factors. Okay, growth factors like what? Like VEGF. Okay, so if you trigger a SASP response in an otherwise senescent cell lineage, those cells are trying to basically turn back on, but they're not just turning on themselves, they're signaling to other cells, okay, including epithelial cells to become to make that transition, okay? When they make that mesenchymal transition, from epithelial to mesenchymal transition, the whole cellular mass from a distance from this initial SASP cell can now turn into a metastatic phenomenon. And this is what's believed to be one of the major mechanisms of why aging is associated with increases in the susceptibility to tumorigenesis. And because now you've got basically a microenvironment making pro-inflammatory cytokines, okay, plus growth factors, and they're going to be acting in now a paracrine rather than an autocrine fashion. So EMT, this epithelial mesenchymal transition, is the emerging mechanism leading senescence or aging to cancer. Been shown that senescent fibroblasts can promote tumor progression and tumorigenesis by pre-malignant cells. SASP can also impact the differentiation status of tumor cells themselves because they're generating growth factors, for example, and pro-inflammatory cytokines. And so they all induce further this epithelial mesenchymal transition. Okay. So it's all very bad news. So as a person ages, all of this can then facilitate tumorigenesis. Okay. So obviously VEGF blocking there would probably be very good. So this puts it all together. This is from a paper in Oncology published very recently, 2017. There's a full citation. This is a open access paper, by the way. So I didn't put the link, but if you Google that in, you get the whole paper. So are there new targets in anti-angiogenesis? Yes, there are. In order for us to understand them, let's take a look at this diagram. So remember, we have monoclonal antibodies. Many of them are blocking particular ligands, okay? Ligands that bind to the epidermal growth factor, to the HER. Remember what this one was? We talked about the triple negative breast cancer. Remember, this is in normal breast cancer. HER is expressed. It's one of the 3 receptors that's expressed. Here's VEGF. Here's bevacizumab. Remember, it's blocking that. Remember, these monoclonal antibodies is what they're doing. Here is another compound. Here is blocking both VEGF and VEGF A and B, excuse me, as well as PGF. Okay, all of these are going to be blocking the reception of those ligands into the VEGF receptor. So you've got crosstalk between VEGF receptor 1, VEGF receptor 2. All this is getting inhibited by these monoclonal antibodies. So you have multiple co-founders of armamentarium blocking various receptor tyrosine kinases, all of which, because these are all growth factors, every one of these are growth factors, are potentially tumor cells, okay? So you're going to be blocking it. So these are all new monoclonal antibodies are in the armamentarium. Also, as I said, we also have, I also said in previous talks, I talked about compounds like nivolumab, which blocks the programmed death receptor, so that even though the programmed death ligand is being expressed by tumor cells, it's blocked to bind to its receptor. So again, this is maintaining T cell viability, right? So you've got 5 different monoclonal antibodies here, all functioning, and here's another monoclonal antibody also binding on this VEGFR receptor 2, this tyrosine kinase. So you've got multiple monoclonal antibodies that work here. All right. We also have the inhibitors that we talked about, inhibitors that are specifically blocking the receptor-mediated responses downstream from ligand binding. And now we're also talking about what the downstream events can be, blocking all of them. Phosphatidylmethylmalonyl 1,3 kinase, AKT, mTOR pathway, the RAS and the BRAF pathway. Again, I talked about this in previous talks. BRAF turning on MEK and ERK. All of this, right, normally would do what? When this is all turned on, if it's not blocked, it's going to cause an increase in nuclear gene expression. It's going to cause cell proliferation, survival, and angiogenesis. It's going to shut down apoptosis. So we want to block this downstream stuff. Let's go back to here. An examination of the canonical oncogenic pathway suggests other targets then. Downstream from all these receptors and downstream from the ligands that bind to the receptors. Okay, so this is all immunotherapy up here. Now these are all just low molecular mass inhibitors down here for these enzymes. What enzymes might we want to block? Phosphatidylmethylmalonyl bisphosphate 3 kinase, okay, because that's going to start this cascade. AKT, inhibitors for that. And of course, back up here, the epidermal growth factor and BRAF, okay? So multiple valencies of drugs attacking at the immunotherapeutic level and also at the small molecule inhibitory level. Okay? In the reversing this process, decreasing nuclear expression, decreasing cell proliferation, decreasing survival via, for example, autophagy, decreasing angiogenesis, anti-angiogenesis, and therefore upticking apoptosis. Okay, so these are core signaling transduction pathways in cells, and they're all linked to tumor angiogenesis. And so, and they all have upstream effects on VEGF, because remember, VEGF itself is one of the genes that gets expressed, right? Remember, it acts in autocrine fashion, right? So blocking any of this is going to also decrease the amount of VEGF generated in these tumor cells. Right. Let's summarize all this. Where are we at? Okay, it seems like we're in a complicated situation here. We are. But the point is, we now have a lot more information of what's going on in tumors than we did even like 5 years ago. So what can we say? Rather than univalent targeting or monotherapy for angiogenesis, combinatorial pharmacotherapy is the new direction. Tissue specificity And temporal delivery to malignancies have to be examined for each new drug combo. Okay, temporal meaning how old is the patient and how large is the tumor cell mass. Okay, so there's a time differential here, which is being fitted into our randomized control trials. Combine that with receptor tyrosine kinase inhibitors and the anti-angiogenesis antibodies and the programmed death ligand immunotherapy. All of that together are becoming promising strategies. Okay, if you hear brand new talks that are being delivered by about multiple cancers in humans, this combinatorial strategy looks like it's working because it's so multivalent, right? Of course, we also always have surgery and radiation therapy and classical chemotherapy, and those are going to continue to play vital roles in anti-tumor medicine. So we're not out of the woods yet, but we're starting to understand that we have to look at specific tissue masses. We have to determine whether or not cells are senescent or not senescent. We have to account for this transition from epithelial to mesenchymal so that you can generate basically cells that can turn into metastatic systems. We have to be careful of that. We have to watch out for the SASP system. And that means we have to be looking at the host itself, the patient, the age of the patient, and also the age of the cell mass that we're trying to ablate. So, all right, so we're at the end of the talk, and I think I made it shorter than usual. Thank you for your attention on this. I guess it's called Black Friday. This is my dog, Luca, and Luca is around all this fur, right? This is all either Douglas fur, these really large rounds here, and this is white fur. So combined, this makes for really good fire in the wood stove. At any rate, I'm Dan Guerra, and I'm from VerEvMed. Here's my personal email address. Please send me any email and let me know what you think about these lectures. Any feedback I greatly appreciate. Here is our email address for the company, info@verevmed.com. Here's our website. Please visit it, and please also leave comments there. Remember what we are scientists verifying published evidence in medical biosciences. That's what we're doing. We're science trying to inform the medical community, including patients, including physicians, including other healthcare workers, and basically all kinds of people, including the legal profession that may be involved in examining what's going on with drug therapy in the open market and insurance companies and whatnot. Okay, so let's get ready to stop here. So once again, hopefully this was an informative lecture for you. I've got at least 2 more I'm going to deliver before Christmas, and I'm also got a couple of surprises of what I'm going to talk about, which are quite far afield from chemotherapy and drugs used in cancer and the discussion of autophagy, apoptosis, and senescence. I'm going to probably throw in an interesting topic, and hopefully people will like it. So all I have to say at the end of the talk is my normal sign up for now, which is goodbye for now.